Welcome to this lecture on animal behavior. Today, we are going to explore a fascinating topic called kinesis, specifically focusing on orthokinesis and klinokinesis. Have you ever wondered how tiny, simple animals with no complex brains or eyes manage to find food, avoid danger, and survive in a chaotic world? They do not have GPS, and they cannot simply look around and walk toward a safe spot. Instead, they rely on incredibly elegant, hardwired movement rules. By the end of this video, you will understand exactly how these non-directional movements work, and you will see how simple mathematical rules can create complex, life-saving behaviors in the animal kingdom. Let us dive in. To begin our journey, let us understand what kinesis actually is. Imagine you are blindfolded in a large field, searching for a hidden treasure. You can't see the treasure, so you cannot walk in a straight line toward it. Instead, you wander randomly, changing your speed or how often you turn based on the beeps of a metal detector. This is exactly what kinesis is in the animal kingdom. It is a completely non-directional behavioral response to an environmental stimulus. Unlike a moth that flies directly into a flame, an animal using kinesis does not steer toward a goal or run directly away from a threat. It simply alters its overall activity level until it accidentally stumbles into a favorable environment. Now that we understand the general idea of kinesis, let us look at the first specific type, called orthokinesis. Think about driving a car on a terrible, bumpy dirt road. You hate the bumps, so you press the gas pedal to get off that road as fast as possible. When you finally hit smooth asphalt, you relax and slow down. You did not steer toward the asphalt. You just moved fast when things were bad and slow when things were good. In formal terms, orthokinesis is a response where the linear speed of an animal is directly or inversely proportional to the intensity of a stimulus. The animal simply runs faster in a hostile environment, which statistically increases its chances of escaping and slows down in a safe zone. This brings us to an important question. Why is this simple speed mechanism so crucial for survival? Consider small invertebrates like woodlice, also known as pillbugs. These creatures lack the brain power to map out their environment or visually locate a safe haven from a distance. Furthermore, woodlice breathe through gills, meaning they will quickly dry out and die if they remain in a dry, sunny spot. By utilizing orthokinesis, they naturally accumulate in dark, damp places. They do not know where the dampness is. They simply run frantically when they feel dry and stop moving when they feel moisture. Without this hardwired behavior, they would wander aimlessly at a constant speed and perish. It is a beautifully simple evolutionary solution. Having seen how orthokinesis works in nature, let us formalize it mathematically. The concept we just discussed can be expressed with a very simple equation. Here, the variable v represents the velocity or speed of the animal, typically measured in millimeters per second. The variable s represents the intensity of the environmental stimulus, such as dryness or temperature. Finally, the symbol k is a constant of proportionality that depends on the specific species and the type of stimulus. What this formula tells us is that the speed is directly tied to the stimulus. If the stimulus s increases, meaning the environment becomes more hostile, the speed v will proportionally increase. When the animal finds a safe zone and s drops to near zero, its speed v also drops to zero, causing it to stop. Before we move on, let us see this formula in action with a concrete example. We are given a woodlouse with a species-specific constant k equal to 2.5. The dryness stimulus, S, is measured on a scale from 0 to 10. We need to find the speed in a damp area, where S equals 2, and a dry area, where S equals 8. We choose the orthokinesis formula because it directly relates stimulus to speed. For the damp area, we multiply 2.5 by 2, giving a relaxed speed of 5 millimeters per second. For the dry area, we multiply 2.5 by 8, resulting in 20 millimeters per second. Physically, this final answer shows the woodlouse sprinting 4 times faster in the dry zone, ensuring it quickly escapes back to the damp safety. Let us visualize the mathematics we just calculated. Here you are looking at a top-down diagram of a woodlouse's movement path. The arena is split into a yellow, dry zone on the left and a blue, damp zone on the right. Notice the dashed arrows representing the animal's path. In the dry zone, the arrows are spaced far apart, representing the high speed of 20 millimeters per second we just calculated. The animal is rushing. The moment it crosses into the blue damp zone, the arrows become tightly packed together. This represents the speed dropping to 5 millimeters per second. The diagram perfectly illustrates how changing speed based on discomfort naturally traps the animal in the favorable environment without it ever needing to know which way to steer. Now that we have mastered orthokinesis, let us explore the second type of kinesis, known as klinokinesis. Imagine you are playing Marco Polo in a pool. If you think you are far from your target, you swim in long, straight lines. But the moment you hear a splash nearby, you stop swimming straight and start turning around in tight circles, sweeping your hands everywhere. You change how often you change direction based on the sensory input. This is the essence of klinokinesis. Formally, it is a non-directional response where the rate of turning is proportional to the intensity of a stimulus. The animal does not necessarily change its speed, but its nervous system triggers random turns at a higher or lower frequency, depending on how hostile the environment is. Why is modulating the turning rate so effective? This behavior is absolutely crucial for organisms that need to stay within a localized favorable zone, like a patch of food or a dark hiding spot. A classic example is the flatworm, also known as planaria. Flatworms lack complex, image-forming eyes. They only possess simple eye spots that detect overall light intensity. To avoid predators, they must stay in the dark. When a flatworm accidentally enters bright light, it drastically increases its turning rate. It essentially thrashes and bounces around randomly until it happens to face the dark again. Without klinokinesis, the flatworm might just walk in a straight line right out of its safe hiding spot and into the open where it would be eaten. Just as we did with speed, we can express this turning behavior mathematically. This formula shows where the turning response comes from conceptually. The symbol omega represents the rate of turning, usually measured in degrees per second. The variable S, once again, represents the intensity of the environmental stimulus, such as the brightness of light. The symbol C is the turning coefficient, a constant that dictates how strongly the specific animal reacts to the stimulus. In plain English, the frequency of the animal's turns is directly controlled by the stimulus intensity. If the stimulus S increases, the turning rate omega will increase, causing the animal to twist and turn rapidly. If the stimulus decreases, the turning rate drops, allowing the animal to move in a relatively straight line. Let us apply this formula to our flatworm example to see the exact difference in behavior. We are given a flatworm with a turning coefficient C equal to 0.5. We need to calculate its turning rate in a dark corner where the light intensity S is 10, and under a bright light where S is 100. We use the Klinokinesis formula because it links stimulus to turning frequency. For the dark environment, we multiply 0.5 by 10, giving a low turning rate of 5 degrees per second. For the bright environment, we multiply 0.5 by 100, resulting in 50 degrees per second. Physically, this final answer means the flatworm is thrashing and turning 10 times more rapidly in the light, increasing the chances that its next straight movement will carry it back to safety. To fully grasp this, let us look at the visual representation of klinokinesis. Here we have a diagram showing an animal's path across two zones. The left side is a dark gray zone with low light, and the right side is a bright yellow zone with high light. Follow the black path. In the dark zone, the line is smooth and relatively straight, reflecting the low turning rate of 5 degrees per second. The animal is calmly exploring. But the moment it crosses into the bright yellow zone, the path becomes a chaotic, tangled mess of tight loops. This is the 50 degrees per second turning rate in action. By turning randomly and rapidly in the unfavorable bright condition, the animal prevents itself from wandering too far into danger and naturally redirects itself back into the dark. We have covered a lot of ground today, so let us summarize the key takeaways. First, remember that kinesis is entirely non-directional. The animal never knows exactly where the goal is. It only reacts to the immediate intensity of its environment. Second, orthokinesis is all about the gas pedal, changing linear speed to escape bad areas and rest in good ones. Third, klinokinesis is about the steering wheel, changing how frequently the animal turns to avoid wandering too far into danger. Together, these two mechanisms prove that you do not need a complex brain to survive. Simple, elegant mathematical rules are enough to guide these animals through a chaotic world. Great job following along, and you now have a solid understanding of non-directional animal movement.